Hi, we're going to talk about experiments and quasi experiments today. And the book seems to think that their quasi experiments are really different than experiments, but um, you know, from a research design perspective, we're talking about shades of gray here. So we're going to talk about those. The biggest thing for you guys to think about are that each research design has a different set of concerns attached to it. In other words, there are things that we can do in the setup of our experiments or the setup of our research design that will really greatly impact whether or not we're going to have strong internal validity, that the relationships in the models that we're building are accurate, or if we're missing something. External validity, how much can we expand or generalize to the population what's going on, from our research. There are some artifacts that might take place, like maturation or compounding effects, regression back to baseline that may or may not take place that we need to be at least aware of. We need to be really concerned about causality too. So if we want to say that something caused something, we need to hit those four criteria. And not all designs can do that. Other things are like how much can we control and how much do we want to not control. Control is really great because it allows us to focus in on the really, really specific sets of relationship between independent and dependent variables. But also what it does is it minimizes our ability to generalize. So sometimes instead of control, what we do is we increase the amount of variation or randomness. So we introduce randomness instead of try to minimize that. So how we do control is important. And then power. Power is this concept about how much strength do we have from the number of cases we have. How strong can we say that some relationship exists? So especially as you're designing some research, like maybe you're in class experiment, think about some of these things as we start to design. So let's talk about experiments. Experiments are a strategy, and we're going to talk about how those work and why we do them. Experiments can happen between subjects and within subjects. Between subjects is to say, like, we have two different groups. We're going to subject one group to a treatment and the other group to a control, and we're going to compare the two groups. But also, experiments can happen within subjects. That means that we can take an individual and subject them to a treatment and then see if that treatment had an effect. So within subjects means we track subjects and we perform that experiment on that individual and we test to see if that changed within the individual. But between subjects means, we take a comparison between these two groups of people. And we're going to talk about quasi-experiments, and I'll give you some examples of how, thinking about experiments very strictly might limit how we use them. So we're going to focus on how we set them up, concerns about internal validity, external validity, control, power. Then we're talking about overall strengths and weaknesses. This is really crucial. And then how do we apply those things? So why do people do experiments? They do them because they're the easiest way to determine causation. They are the best strategy to figure out causal linkages. There is always a problem of directionality in things like correlational research or big broad case studies. What caused the other thing? Well, in experiments, we increase the amount of control, and by doing so, and we set up a control environment. We subject them to a really specific set of stimulus. We manipulate that stimulus. We can tweak that to see if that causes different sorts of effects in these individuals. We can manage time. We can minimize external influences. There are a lot of things that experiments do very, very well. Key components here are manipulation. We can exactly apply a really specific type of treatment in an experiment. We can manipulate the surroundings. And we can be very, very precise in our measurements because we have everything very controlled. Experiments allow us for really good sets of comparison between subjects or within subjects because we can get really up in people's business. And part of this control problem is actually a part of the reason why we do control actually is really detrimental to experiments, because the more that we control, we might be able to find causal linkages, like this thing caused that thing. But what it does is it reduces the amount of similarity between what's going on in our lab and what goes on in the real world. So we reduce the amount of generalizability because we've controlled for all these external factors. And we know that we don't live in an experimental lab. We don't live in a vacuum, right? A vacuum. A vacuum is essentially like we're going to reduce the amount of air. We're going to reduce the amount of, surface tension. We're going to, reduce all these other effects. So things behave differently in vacuums, right? Than they do in the real world. Things behave differently inside experiments than they do in the real world. That's because we've added all sorts of control to minimize external influence. So in terms of manipulation and control, we're going to control the subject's exposure to the independent variable. We can control the independent variable. We can not subject them to the independent variable. So let's say if we're trying to figure out if a certain sort of growth hormone or certain sort of like chemical affects neural functioning, we can add smaller amounts, we can add bigger amounts, we can withdraw that, we can just manipulate all sorts of levels of that independent variable, and we can measure the outcome pretty directly. That's pretty awesome. We can also minimize control, or we can minimize exposure to other factors. That's part of control too. We can minimize environmental variables. We can control who gets to participate in the experiment. Those are participant-related variables. We could screen for people who are, 40 to 44, who are bald and wear glasses and, talk funny with their hands all the time, and only study those people. And then, not study anybody else. We could also manipulate who gets to be into our sample by paired matching. A guy who's 205 pounds, we match him with another guy who's 205 pounds. They're matching in terms of hair color or hairstyle or baldness or, glasses color. We can pair people together and subject one group to one type of stimulation and the other one not. We could do this in public programs. Like we set a family of 4, we get another family of 4, we, pair them up as much as we can, and then send one group through a government program and the other group not through a government program, and see whether or not the government program helps them. So experiments are a lot about manipulation and control. We can also control time-related variables. How long are they exposed to the independent variable of effect, like to the treatment? We can cut off the amount of time. We can use time in terms of regression back to a baseline condition. So let's say we give someone a stimulant and they're all jacked up for some period of time, and then we can wait before we give them a stimulant again. Or we can, halfway through their time that they're crashing because they drank way too much caffeine or something, we could give them another set of stimulants to see if there's a secondary effect or if there's a reduced effect, or what the true time, the true maximum capability of that substance is. So we can manage time, we can manage duration of exposure, we can do all of those sorts of things inside of an experiment. One of the control strategies is holding a variable constant. We're going to keep the temperature the same. We're going to keep the location the same. We are going to, keep the amount of exposure the same. We're going to keep a certain type of subjects variables constant, like only weight. We're only going to measure these people who are at a certain weight. We're only going to look at a certain type of species or a certain type of exposure. That's like holding one of those things constant. The other way to do this is to have some sort of control is matching. We talked about that. Matching 2 different types of subjects. We could match 2 different types of locations and subject one location to another, one stimulus and the other one not to a stimulus. So we can match different characteristics to try and, more or less control what kind of participants we have, or match sorts of exposure if we're out in the wild, like some sort of exposure to sunlight by 2 different types of location by matching those locations and then treating them with something else. Another type of control strategy, which I kind of alluded to, is called randomization. And we can control through randomization if we have a large enough sample. That's to say, if we can get enough people and we randomize them into different groups, or if we can get enough subjects that have a variety of different characteristics, and when we randomize them into 2 different groups, then we don't have to match, because the natural variation within these groups, if they're large enough groups, will mimic each other and the real world. So in randomization, as long as the groups are big enough, we don't have to do like control by matching or by holding some variable constant. Now, Randomization does require big sample sizes, but it also assumes that those samples aren't biased and that the variance between those samples, the differences that exist in those samples, is similar between those groups. And that can be difficult if you have a really small sample size. So randomization is one possible way to use control, but it's not always the best way, especially if you're doing small samples or if you have really two distinct groups and their variances are not similar. Okay, another way that we do control is through control groups or non-treatment groups. And when we do non-treatment, people know that they're not being treated. So we have to give them placebos because the placebo actually has effect. Giving someone a pill gives them hope, and then their bodies react differently. I was in a control study, and I wanted to lose weight in graduate school, so I was like, "Sweet, sign me up for this weight loss program." And it was an exercise study. I got selected into the control group, and I was frustrated because I wanted to lose weight. That's why I was there. But then they said, for the next 6 months, you just have to eat like normal. You can't exercise any differently. In fact, don't change your life at all. You're in the control group. And I was like, what? And I gained weight. You know why I gained weight? Because I had this motivation to go, and then they told me to like not do anything. I'm like, well, how does that help? Like, they should have at least allowed me to just live my life the way I wanted to live my life. Maybe I would have lost weight, maybe I wouldn't. But to tell me to do nothing actually accentuated the negative effect of being in the control group. So you have to think through how being in a control group might affect someone. And then there's some ethical concerns about these experiments. For example, my mom had breast cancer, and she was in a study because I think when people have life-threatening illnesses, they'll sign up for sort of any type of potential cure. So she ended up at the James Cancer Institute at Ohio State University. They put her into a study to look for the effectiveness of a certain kind of drug at treating, but also treating nausea. But if you were in the control group, you couldn't have the standard anti-nausea treatment. And she said, like, about 2/3 way through the study, she just gave up. She gave up because she knew that she was in the control group. She knew that she could have some anti-nausea medication, and she was just suffering for months, knowing that there was a treatment and knowing that she was in the control group, not getting treatment. And she's like, fine, forget this, man, I'm out. So when you have people involved in control groups, they tend to fall out if they know that they're in the placebo group. And I think, like I said, there's some ethical concerns. So you have to be careful and cautious how you do treatment and non-treatment. Placebo is the easiest one to do for humans, but if you are visiting like a wild site and you're looking for birds and you're looking for these different types of strategies, if your normal procedure is to go and visit those places in the treatment group, you have to go and visit those places in the control group too. And you have to make the sort of same disturbances as an observer you would in both of these areas, because even though it's not part of your study, you walking into that area has an effect. So even though we say like, well, that plot's the control plot and this plot's the treatment plot, let's go look at the treatment plot. Well, that means you got to sometimes go and look at the control plot too, because you're interfering in that experiment by not being there. So it's important in an experiment to treat all of the treatment groups, treatment or control groups, the same, and minimize the differences in what you're actually doing. Okay, experiments are so good for internal validity. That's to say, does this relationship exist? Does this independent variable affect that dependent variable? Like, does this independent variable affect that dependent variable? You can look at all these little things that are, affecting some sort of outcome. That's very cool because we model internal validity, like with this one does cause an effect, this one doesn't cause an effect, and we can minimize the number of those external variables to try and figure out which one of those is actually causing a change. Except that when the level of control increases, in other words, as we control for more and more and more things, it shows up that experiment looks less and less like the real world. Now, in terms of generalizability, that's external validity. Would say, well, then we can't extrapolate what's going on inside this experiment to what's going on in the outside world because we've controlled all these variables. But also, we might have internal validity problems where some variable might be interacting with another variable to cause a change. And we're not measuring that because we don't have that second variable. So, I think about like drunk driving and caffeine. So when they're making these like Red Bull alcoholic drinks, the caffeine is keeping you jacked up, and the alcohol is slowing down your sensory responses. So if we're looking at sensory response, and we're just giving people alcohol, totally cool. If we're just giving them Red Bull, that might be cool too. But the interaction effect between those two causes some totally different kind of outcome. Now, if we're trying in an experiment to control, to see whether or not alcohol has some sort of effect, but most people aren't drinking just alcohol, they're doing both of these things, the Red Bull and the alcohol, then we've controlled for that. And we can look at the effect of alcohol on sensory reaction time, but we might be really missing out on a larger part of the process that's going in there because we tried to control for that. So we do all sorts of things. Like when we do an experiment, what we might do is a simulation. We might do some computer modeling, or we could go out into the field to test whether or not our experiment holds up, the results of our experiment hold up in the real world. There are different ways that we can sort of make sure that our experiments are internally valid, but then also externally valid. So even though they exist inside of a lab, it doesn't mean we stop out, in the lab. We go out and test them if they exist in the real world too. Experiments generally can be broken up into two classes. One is between subjects, and the other one is within subjects. Between subjects are different individuals, different groups of people, and we give them the same treatments, or we put them into a control group. And there's one score of the dependent variable per subject per time period. In other words, we measure some outcome of that individual per time period in that group. And we measure some variable of that person in that group per time period. We are not tracking individuals over time in between subjects necessarily. The experiment itself relies on a comparison between a control group or a different treatment group, some sort of other treatment. Control can also just be a non-treatment. So non-treatment, treatment 1, treatment 2. What we're doing in a between subjects is comparing these different groups who are given different stimulus. And we're seeing if there's a difference between each group of these subjects. Okay. A within subjects experiment takes an individual and tracks them over time through multiple types of treatment. Maybe that's a non-treatment in time period 1, and then we see if there's a change in time period 2. In time period 3, we're given a certain type of treatment. In time period 4, we take a measurement. In time period 5, we give them a treatment. Time period 6, we take another measurement. And we're tracking like Justin in April, and then we give him some drugs, and we see what it looks like in May. And then we give him some drugs, and in June we follow up with Justin, and we're seeing whether or not Justin changed over time. That's a within subjects. Is the change happening within that individual? There are multiple scores of this outcome variable, the dependent variable, per subject. Most often it's after some treatment or a return to baseline. This question about within subjects we're probably going to come back to, but sometimes we stimulate a subject, we give them some sort of treatment, and they never return to baseline. So in a within subjects treatment, we can subject Justin to like a punch in the face, and we measure how much it hurts. And then the next time we get ready to punch him in the face, my reaction is going to be dependent on the fact that I also was hit in the previous time period, so I might flinch more. And then my muscles will be tighter, and I don't know if that's going to hurt more or if it's not going to hurt more. But I never return to the baseline because the baseline is, I don't know what's going to happen. Oh my gosh, that just hit. Like that sucks. I will never go back to, I don't know what's going to happen again. I know what's coming next, and so I can't return to the baseline. Same thing with sort of like drug addicts. There's some drugs that like fundamentally change the chemistry of your brain. So like methamphetamines, let's say, for example, or crystal meth, or whatever it is you want to say, like once you do that one time, something changes in your brain, you like the release of that dopamine or whatever it is that happens. If you're given the chance to do that, your body starts anticipating that and what that reaction is. And so you never really like come back to your former state. So that return to baseline is really, really important within subjects. And if it's not a return to baseline, what we have to do is we have to randomize throughout the group who gets which stimulus when, and we have to randomize treatments to make sure that even if they don't come down to baseline, they can that we don't get additive or cumulative effects that would be biased. So we have to randomize the different types of treatments so we don't accumulate bias in a certain type of pattern of giving stimulus A and then stimulus B, then stimulus C. Some group gets A, B, C, some group gets B, A, D, some group gets some other sort of set of treatments over time so that we can fix this problem of returning to baseline. All right. Sorry, I got off on a tangent there. Between subjects designs, between two different groups, individual differences, how did one group get into one group or the other? This is a confounding variable. So if we're going to run an experiment, we have to make sure that we're truly randomizing or if we're matching. Now, for matching, we have guaranteed bias, but we minimize the guaranteed bias by selecting on-set characteristics and assignment. So it's bias that's known to us. When we randomize people into different groups, we have to have a big enough sample to have true randomness among the groups. And if not, how did they get assigned to those different groups? And are there some unknown biases in how they got assigned? One of those assignment biases, let's say we take the first 10 people who sign up and we put them in one group, and then it takes 10 hours to get the next 10 people to sign up for whatever that study is. The first group are really willing participants, and the second group were like, yeah, maybe we'll do it. The first group has a set of characteristics that made them sign up that's faster than this group that had a set of characteristics that made them a little bit slower. So those groups are kind of messed up. So we might have to say, well, group, as we show up in line, you're assigned this way, you're assigned this way, you're assigned this way, you're assigned this way. So we just have to make sure that as we set up these groups, that either they're randomized or they're matched, or in other words, we can minimize assignment bias. Okay, because if we have some bias in how those groups were set up, the comparison between those two groups will be biased no matter what we do. There are other things like environmental variables. Did one group get subjected to some sort of stimulus that the other group didn't get subjected to because of where they were or what they were exposed to, what they saw on television, where they were located on a slope? There's all sorts of things that could affect how these two different groups experience the world differently. And those are called environmental variables. And so we have to minimize as much as we can this some unmeasured environmental variable that might have an influence on how these groups are compared to each other. So how do we limit these problems? We create equal groups. We treat each group the same. We make sure as much as we can that each group is comprised of equivalent individuals. How do you create an equivalent group? Random assignment with some large sample? Sure. Or matching. Now here's a question, how do you match participants and on what characteristics? Because nobody's the same, but what are the key criteria and how many criteria are there to match an individual with another individual? Do you have to match on gender or race? Do you have to match on height or weight? Do you have to match on, some other genetic predisposition? Well, it kind of depends on the question you're trying to pursue. No matter what you do out of this whole class, I need you to remember that however we design the research should be dependent on the question that we're trying to ask. If we ask a certain kind of question and it leads us to an experiment, awesome. Then it should also lead us to, well, how would I match participants? What are the key criteria that are important in matching? And what criteria can I throw out? That should all be driven by your problem, by your research question. All right. So once again, minimizing problem. We treat everybody the same way. We set up standard procedures. And you say, well, is that really hard? Totally. It's hard. You have to show up at the same time every day, or you have to show up in a certain pattern. You have to come back and use the same words, or maybe you have to, progress through the field in a certain way that's exactly the same. Well, why does that matter? Well, because your procedures have to be so standardized that a change in the procedure doesn't introduce a new stimulus inside your experiment. Because changing the procedure will make those two groups not the same if one group gets done one way and one group gets done another. So, we talk about experiments, like these are very formalized ways to look at the world and to test hypotheses. What we're trying to do is minimize the amount of external influence so that we can maximize our internal validity to see if, what's causing what. All right. Another way to minimize problems is to take a huge sample and randomly assign people. So instead of matching, instead of, all these other things, if we just get a big enough sample that's reasonably random and randomize those people into different groups, then the natural variation in those groups should create more or less equivalent groups that aren't based on like some set of criteria that you chose. It's just based on natural randomness. So we kind of talked around some of these twice, but there are ways to minimize problems. You just have to kind of think creatively, like what could introduce bias? How could I overcome that bias? What do I know about the laws of probability? What do I know about design? What do I know about potential problems so I can minimize those problems? Okay, in experiments, especially between subjects, you have major problems. Differential attrition is where one group drops out faster than the other. So like in the example I gave about my mom and the breast cancer treatment, the experiment treatment, most of those people stayed on because they had a doctor who was monitoring them constantly about their breast cancer. The control group, they experienced different feelings. There were some significant hardships. They had additional health problems, but the doctor was unable to convince them that they needed to stay because the doctor is also in this weirdo position of saying, well, if you drop out of the study, I can actually help you. So what happens is this study with the experimental treatment stays like 98% of those people stay, but 50% of the people stay in the control group because they realize they're in the control group. So when that happens, you have to say, well, I've got 100% in this group, A, and 50% of the people left in group B. How can I reasonably compare group B with group A? Because there might have been some process that was going on where the people who dropped out had a set of characteristics that now this group doesn't have. So in other words, the ones who stayed in the control group that my mom dropped out of, maybe they were stronger, more physically fit, maybe more psychologically, ready to deal with cancer than everybody else. This group over here has those people too. But they also have the people who are not as psychologically strong, not as prepared to deal with cancer, blah, 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 blah. Those people didn't drop out, but these people did. So when you compare them, those groups are no longer the same. Differential attrition happens also when subjects die. So it's possible that this group, and I was talking about, once again, breast cancer treatments, it's possible that more people die in this group than this one. Well, the people died out here. How do I compare? These people are still alive. These people are still alive. Had these people not given the treatment, they would probably also have died, but they didn't. And now these people did die. How do I compare the groups? They're no longer the same. Well, if there's some process that's causing attrition inside your groups that's biased, then now once people drop out of those groups, your samples are biased. Your comparison groups are not equivalent. That's called differential attrition. Now, there's also this thing called diffusion of treatment. If these two groups are able to interact with each other in some way, there could be a rivalry that shows up. There could also be equalization. They could be sharing tips or tricks about how to overcome their problems that are going on. They might share what's going on with their treatments. The hope that one group has might spill over into the hope that the other group has. We might actually get, especially on like psychological experiments, we can get equalization where some of the benefit actually transfers to the control group because of hope or because of something that's going on, like in exercise. They say, oh, well, you should try this thing. Okay, well, cool. Well, then now these guys have had a sort of secondary treatment because this group who's being taught to exercise says, oh my gosh, my friend's in this other group. I should tell him too. Well, he's getting treatment, then talks to his friend in the control group about how to do something better. then now we have kind of a contamination, and they start talking, and then they've diffused like a secondary set of treatments. The other thing that can happen is that these two groups could be demoralized. One could be demoralized, and there's some psychological things that take place as one's getting treatment and one realizes they're not getting treatment, that compounds the effects of the treatment or non-treatment, so that it's not just a simple treatment or non-treatment. Now there's some like emotion attached to that. And you say, well, that doesn't really happen with, I'm not interested in humans. It doesn't really happen with animals. Totally does. Totally does. It happens with plants too. I mean, we talk to plants and they grow better. And we say mean things and they don't grow as well. I think there are a lot of things that we can just like write off and say, well, nature doesn't really work that way. Well, we're finding out that nature really does. Like, kind of have feelings, and you're gonna think like I'm kind of stupid about this, but we often don't understand a lot of these things. And so in an experiment setting, we have to at least assume that some of these things might happen because we haven't observed them or don't know about them yet. That's why standardized procedures are so important, and trying to keep samples separate are so important. Not just with humans, but with like plants, cross-pollination, or animals going from one location to the other. So those are some potential problems. Now, how many treatments do you give each group? Well, simple 2-treatment experiments really oversimplify reality. Like, let's say we're exposing somebody to some new idea about global warming, and some group doesn't get that. Okay, well, both groups have probably heard something about global warming, and that's probably not the last message they're ever going to get. So how powerful is one message about global warming over a very short time period? Probably very, very, very small. Even if it does have a major shift in someone's attitude, like as we present some information about global warming to a treatment group and we don't to the control group, well, that's only one set of treatments. That's one time they saw something. That's really not what reality looks like. Same thing happens in like abusive homes or in really positive homes. It's not like the dad or mom, punches somebody, or on the alternative, says something really nice that one time and it changed that kid's life. Maybe in abuse, it can really, really mess them up. But that's probably not just the only stimulus that's going to have, positive or negative. Because if a parent is praising that child, it probably doesn't happen just once. It happens like multiple times over time. And the same thing would happen, in an abusive home. It probably doesn't just happen once. It happens like multiple times. So when we do this like simplified experimental treatment inside of a lab, and we give it a stimulus once, that doesn't really reflect reality. So we have to figure out like, well, okay, how can we increase the number of treatments? How can we, increase the number of participants? How can we decrease these other problems if the relationships don't exist? So like we have to think about like, oh, well, if we give them some treatment, do they return to baseline? And how can we create these stimuli inside 2 separate groups of people and have those effects more or less try to be the same? So once again, there are lots of problems with experiments, especially as we're trying to deal with 2 different sets of people and comparing them, or 2 different sets of study subjects, whether or not that's plants or animals or humans or whatever it is you want to study. Whenever you're studying 2 separate groups, the between subjects design, you're going to have these problems in some shape or form. And I'm a political scientist, so I talk about people. Now, within subject designs, this is sometimes called a repeated measure design. So if you Google that, it's an experiment where you take a measurement of those subjects over time, or at least repeatedly at some interval. Now, what this does, and the power of a within subject design, is different than a between subject design. In a between subject design, we had to make sure that the groups were more or less equal, whether or not that was through randomization or through matching. What we're trying to do is get these individual groups to kind of look the same. Well, in a within subject design, you don't have that problem because that individual is tracked over time. There's no difference in that individual except for maybe what that treatment is doing. So the individual differences are minimized. We don't have to worry about the differences between these groups. We have the same individual under different treatments. Now, what this does is that each person serves as their own control group at a different time period, whether or not that's at the beginning of the study, or whether or not they're given some sort of treatment and they're allowed to return to their baseline, and then they're given some other treatment. Like at some point within subject design, that individual is their own control group. I know what I was like before. Now I've gone through this thing. Now I'm, bigger, better, stronger, different, worse, whatever. Now, the other thing is that it allows for fewer participants because there's a significant amount of control when you're just studying individuals. So all of the variation of the individual all of the external variables that might affect the samples in a between subject design are now confined to an individual. Except we have some additional problems in that each individual may not be representative of the bigger, broader population. So we have to, figure out how to do this to minimize that. Whereas when we had a between subject design, we could gather a huge group of people that are more or less reflective of the population and subject them to these treatments or individuals. Within subject design, you have fewer participants because you're just tracking how an individual responds. But an individual is probably not very representative of the population. So what tends to happen within subject designs is that we have fewer participants, but because we have fewer participants, we have less power. Okay. The bigger problems about within subject design are right here. Time. People get fatigued. Subject sites get fatigued. You'd sound that. I mean, if I'm going out to a forest and I walk into the same plot every month or every day, I'm actually changing the dynamic of that ecosystem because maybe I'm scaring off animals. Maybe I'm increasing the erosive potential because I'm compacting the soil or I'm moving some sort of vegetation off the land. Like that site gets used to me. It sounds dumb, but there are impacts of me constantly coming there. Individuals, whether they're animals or plants or humans, they get fatigued by constantly being stimulated and then being allowed to return to a baseline. So fatigue happens. It can be destructive, but it can also predispose you. We call those testing effects. Okay, history. Something happened before you were there that you haven't measured, and over the course of the experiment within subjects, what goes on in those previous time periods, testing effects, there are prior treatments that have affected them, but also they're predisposed to a certain type of response based on something that happened before you were there. That would be history. Maturation. If you are getting older, like me, you realize that your body just is different over time. What is that? It's just age. Okay, well, how do I distinguish what's age and what's a treatment that occurs every year? You're going to have a hard time disentangling that because over time, maturation within a forests change over time, and they grow to like an apex type of community. Well, how much of that is due to the treatment, and how much is due to the fact that it just does as things age? That's something you got to figure out. Instrumentation is a real problem, especially as we have really good technology. Like we might be able to measure the salinity of the soil with one technique, 50 years ago. And then 25 years ago, we've got a better technique. We can measure it better. And then 10 years ago, we got a better technique. And now we've got like the most cutting edge technique. Well, how do I compare the measurements from before with the measurements now if those ways of measuring have changed, if the instrumentation has changed? Well, you have to figure that out. Maybe you measure in the old way to try and see if it correlates with the new way. Well, you say, well, I'll just use the same probe over time. Well, you can't use the same probe for 50 years because they wear out. I mean, I've been using the same scale in my bathroom for like almost 20 years. Yeah, gosh, isn't that horrible? And I think it's tired of me. Scales used to have springs in them with this dial. And, putting my 200-pound body on that scale every day for, 20 years, the spring's going to wear out. If we start digging soil probes into the soil, they just wear out over time. So you can't use the same instrument. You have to buy like the exact same model, or, like you have to use the exact same chemicals. But what if that chemical manufacturer, if you're doing soil tests, now doesn't produce it in the same way? Or maybe the EPA has changed methods. All those instrumentation problems can really affect a within subjects design. Here's another one too. From a social science perspective, the words that we use now to define or to describe somebody's gender are new. Like 10 years ago, when we asked survey questions about gender, it meant, are you male or female? Or something that's different than that. And now we've got this whole spectrum of words that all have different meanings. But those meanings change over time. And so survey questions that we asked 50 years ago, or even 30 years ago, maybe 20 years ago, they don't have the same meaning, or they don't carry the same social implications as for questions that we use now. But in order to be consistent and measure the same thing over time, we have to use the same words. And if you're using a within subject design and you're trying to measure one individual over time, a group of individuals, but individually over time, your instruments have to have they have to be measuring the same thing in the same way. But culturally, things change. So that can be a real problem. Here's another one. We used to say this, I grew up in the Cold War, and people who were born not too long after me, they don't remember being afraid of Russia and nuclear war. And then 9/11 hit, and I'd say, well, there was a time period before 9/11 and a time period after 9/11. And, the post 9/11 world is just different. It's just different in all sorts of ways. Well, how? Well, it's hard to describe. There's like a whole bunch of these things that are just different. These are confounding variables. Like there might have been something go on in the world that was so big that it just fundamentally changed what's going on in the world and how people react to that, or how study sites react to that. We're going through one right now. Like we had a pre-pandemic world, and now we have a post-pandemic world, a pre-COVID world and a post-COVID world. Well, when we ask people about their satisfaction with education in 20 years from now, that is going to be tainted by the fact that they experienced this COVID time period. And the people who don't have any exposure to education prior to the COVID, they aren't subjected to that necessarily. But within subjects, we say, well, how did that individual or that site or that study, whatever it is, change over time? There are confounding variables that just make those time periods different. And you can't really assign that difference to just a set of confounding variables, like it's, pre-COVID, post-COVID, but it could also be part of the treatment, you have to just be aware that individuals might collectively experience some event that really changes how they see things, or how they're affected by some treatment. Other things like environmental variables that we can't track, that are outside of our control, because we can't just hold subjects in a lab for days or months or years. They have to go out and experience life. But all those people within subjects, they're having different experiences out there. So that's hard. And then the other thing is attrition. Attrition is a problem for everything that deals with time. Between subjects, we're talking about like two groups and who's dropping out of groups. But within subjects, attrition could be like, yeah, that person died, or that person decided to not show up anymore. If we're tracking like people in probation or parole, they're not required to check in once their parole is over or their probation's over. So like, what do they look like 5 years after parole? We don't know. But because they had to show up for a routine check-in during parole, we can measure them over time. Once they're no longer required, they're not coming back. So, our studies are limited. Some of the bigger problems within subject design are carryover effects and order effects. And the best way to describe this is to say, I have two individuals, and I have like 3 different types of treatments. The order in which I give someone these treatments may have an effect. So if I give them treatment A, and then I give them treatment B, and then I give them treatment C, something might have happened in the A that affects the way that B is received, that affects the way that C is received. So what I have to do for within subject design is these 2 different individuals have to experience the order of the treatments differently. So this one gets A, B, C, this one gets C, B, A. And then another group gets B, A, C. And the order in which they're given that stimulus gets washed out because they all get a different order. They're randomized in the order in which they receive the treatment. That's one thing. Now, we could also have a carryover effect, and a carryover effect is, let's say we have a treatment A, and it's like some sort of drug that lasts in your body for 3 months. Well, if we give them treatment B 2 months in, they still have another month before treatment A goes away. So they never really came back to a baseline condition. They got a stimulus, and then they returned part of the way. But now, instead of being here where they initially were at the baseline, they got a stimulus. Now they're coming down. They never get to baseline. They only get to here. So the difference between the baseline and where they were when they got the second set of treatments could be really problematic. So they have to return to a baseline condition. So you have to ensure within subjects that if they're given a treatment, that they return to normal before they can be given a second treatment, like in order. So we don't have compounding effects of these treatments. And you think, well, you can talk about that with humans, like whatever, no big deal. It happens with trees. Like if we go out and we treat a tree and it causes that tree to more or less fundamentally change its nature, it will never return to its base state. Or if we're out so we can never give them a second stimulus or another treatment. With the COVID vaccine, they want to look at how effective one shot is. But if you don't get the second shot, like how can you, we would never not give somebody the second shot. So we give somebody the first shot, and it's like 80% effective or whatever. And then when time rolls back to give them the second shot, they've never recovered from the first one because now they're immune, like they've been immunized and they haven't returned to a baseline condition. But we give them the second shot anyway because we know it's going to work, right? We wouldn't leave somebody unvaccinated just to see whether or not they get it, to see what that rate was. And the booster shot, that second vaccine shot, helps protect them more. But we only know it helps protect them more because they never return to the baseline to see what the individual impact of that second shot would look like, as if they had never received the first one. Anyway, with trees, we do the same thing. Let's say we want to see, the effect of a certain type of treatment, to grow a tree. Well, okay, we grow the tree and it's got some treatment in the soil. Now the tree's bigger. Well, bigger trees tend to use more water, but also they grow at different rates. So we can't go back and sort of return to the baseline condition to see if a different treatment will affect them differently. So within subjects have these order effects and carryover effects, and we have time and environmental. So they're a really good alternative to between subjects, and between subjects have lots of strengths, and within subjects have lots of strengths. So one of the things that we can do is called counterbalancing, and what it does is it minimizes the effects of carryover and as well the order effects of treatment. So it prevents any order effects from accumulating in one particular treatment condition. Instead of the order effects are spread evenly across all the different conditions, so it's possible to make fair and unbiased comparisons between treatments. That's on page 261 of the book. Okay, we talked about that. We talked about randomizing these different treatments. The most common of these is called a Latin square. You can Google that, and it says, this group is going to, receive the treatment in this way, and this group's going to receive the treatment in this way, and this group's going to receive the treatment in this way, and we randomize them so that they're kind of spread out. If you wanted to really dig in, you could look at comparing the within and the between types of experiments. There's a table on page 267 of the book. The strengths of one are the weaknesses of the other. There's individual differences or there's group differences. Time and order related effects aren't really a big deal with between groups, but they're a really big deal within groups. The number of participants it takes to get a really good, reasonable representative sample with between groups, you don't have to deal with within groups. There are individual differences, problems within and between. No matter what, the kind of question that you ask should drive what kind of research you're going to do. So there's this big distinction out there in the world between experiments and quasi-experiments or non-experiments, blah, 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 blah. Let me just explain to you why I think that's a big load of stuff. The main factor that distinguishes experiments and quasi-experiments and non-experiments is that experiments are designed to establish a cause and effect relationship. And they are designed to remove confounding variables. In other words, minimize external influences, conduct your experiment in a highly controlled, highly manipulated environment, determine causation through manipulation, through time order, et cetera, et cetera. And people say, well, so if it looks like an experiment, but it's really not controlled as much, we'll call it a quasi-experiment or a non-experiment. And I'm like, well, can you remove all of the confounding variables in an experiment? Maybe, but probably not. So I think really what distinguishes them is that you can limit the external influences more in an experiment than you can in a quasi or a non-experiment. In other words, the degree to which the strategy limits confounding variables and controls threats to internal validity is what distinguishes them. But I think that if you're a good researcher, you're going to try and limit problems, whether or not the book calls it a quasi-experiment or a non-experiment. You're going to try and figure out where you got these problems. And if you call it an experiment or quasi-experiment, I don't really care. The idea in experiment is like separate groups, try to control, try to manipulate the stimulus so that you can see if there's a difference either within or between. Okay, so I think about experiments as a category of research designs. Talking about quasi and non-experiments, sometimes we have to examine pre-existing groups. We don't have the luxury of separating them into randomizing them into groups. Like if we do happen to use pre-existing groups, we have to use those. How do we deal with that? Well, we have to recognize that there's probably assignment bias. Like we didn't get to select who got into which group, so we assume that there's bias there, and we try to minimize that as much as possible. One way to do that is increase the number. If it's a big enough set of two groups, then we probably can assume that they're done by randomness, or we can go back down into the data to figure out how they were assigned and see whether or not they meet some reasonable standard. Even when you have some bias in assignment, you can determine whether or not it's going to be influential. Here's another one. It's a non-experiment technically, but it's an experiment anyway. So it's kind of a quasi-experiment, and it's done through my research. It's called a conjoint experiment, and it's embedded in a survey, and it's part of this book chapter called Who's in Your Lifeboat? Now, during the early on in the COVID pandemic, about May of 2020, I said, let's launch a survey. And we surveyed 2,000 people, blah, 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 blah. We asked them a bunch of questions. You'll see some of these results in another setting. And then we randomized what they saw. They either saw a news video clip of someone who died of COVID after exposure at a barbershop, or they saw a very similar length video about Black Lives Matter protests from the same news source. We did a pre-post treatment. You won't see those results here. Really what happened was we were trying to see if people's perception of COVID would change if they saw a sympathetic story or whether or not they saw something that was not sympathetic. So we talked about the Black Lives Matter one because the person who died of COVID was black. And so we tried to minimize the differences between the treatment and the control. So in the treatment, we had a 42-second ABC clip of a black person talking about COVID. In the control, we had a 45-second ABC clip of a black person talking about Black Lives Matter. Much as possible, we could minimize that. Ultimately, that sympathetic story had no effect. So you're not going to see that. But what we did do that did have an effect, and you'll see this in the correlational research, is try to figure out what was related to perceptions of government support for policies. Move that aside for a second. The one I want to talk about now is in that same survey, we did what's called a conjoint experiment, and we subjected these people to having to choose who would they save if they could save anybody. So we gave them a choice of two different people, and we randomized the demographic and group traits using, it's not truly randomized because we the way that the program works, they pair up a number of these, and then the statistical analysis matches what was the determining factor between the choice as you do this. Anyway, so what that looked like was if you had to choose someone to save from COVID, who would it be? And we're looking at in-group, out-group pressures, race, ethnicity, religion, politics, and how much they knew. So we randomized who was between these two, what their gender was, what their race was, what their age was, their party affiliation, their religion, religiosity, gun ownership, and ideology. What's that look like? Over here on the right-hand side, this is the prompt we gave them. Conjoint experiment, if you want to look at that, Heim, Mueller, Hopkins, Yamamoto. You force them to choose who they would save. And then we do this AMCES to figure out, on average, who chooses what based on their demographics. So you can read through this if you'd like, and you can pause it and check. But this is what they saw on their survey. So we didn't randomize who was in the survey. That survey was nationally representative. But what we did was randomize the groups of people that they had to choose from. So in each of these, they got a typical person, a stereotype. I don't even know what the right word is, but they got the description of these two individuals, and the description of those individuals was randomized based on what their gender was, what their race was, their age, blah, 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 blah. Who would you choose as a priority to save? And because we did this 3 times with 2,000 people, we ended up with 6,000 choices. And what we found was this. If we looked at the complete data, the 0 line here is where there's no difference between individual 1 or individual 2. So in the complete data, the only way you see a relationship is if this line does not cross the middle, then there's a relationship there. So in here, no relationship on gun ownership, no relationship on early age, but they tended to favor would be on the right, people who were older, 65 to 75. No difference male-female, no difference conservative-liberal, no difference, blah, 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 all through these demographics. So on average, what was going on was that people were choosing to save older people in the complete data. Then we went through and we started to say, well, what do white people want to do? We went, okay, well, white people, no. Gun ownership, no difference here. Oh, look, they wanted to save old people too. And no differences, none of these. Oh, and look, white people tended to save strong Democrats instead of strong Republicans instead of strong Democrats. What? Yeah, that's what the data bore out, that white people tended to save strong Republicans. Okay, well, that's kind of interesting. Then we said, what do strong Democrats, who would they save? Well, not gun owners, no difference there. They're not saying don't save them. They're just no difference. They don't not save gun owners. Now they tended to really closely to save this, but it's not significant. They tended to save old people, yep. And then no other group that they prefer. So all of the strong Democrats, people who affiliated that way, didn't tend to save anybody except for old people. If we ask them in terms of liberal, not in terms of their party, but their ideology, they don't end up tending to save anybody different or disadvantaging anybody different except atheists. Liberals tended to save atheists over evangelical Protestants. That's kind of interesting. Well, what about strong Republicans? Strong Republicans, they didn't tend to save gun owners. They didn't tend to save old people or young people. They didn't tend to save anybody. Really, there was no pattern among strong Republicans about who they would save. All right, now if we say, what is your religious identity and, let's say, like weak Republican? So we've run, all the variations here. If you're a weak Republican and strongly religious, nothing, they were less likely to save Asian people. Huh? And then nobody else. They didn't save old people. They just didn't want Asian people to be preferred in terms of saving. So my co-author and I said, well, why is that? And so we went back into the data a little bit more, and we found some really strong support that says the way in which the president and some other people were talking about this being a Chinese sort of flu problem. People were like, well, I'm strongly religious and I'm kind of weakly Republican. More or less, they tended to believe that people who were from Asian descent should not be favored for saving, in part because of where the virus came from. at least that blame attribution. Okay, did the same thing. Evangelical Christian, strong religious identity, they tended to not save anybody, not even old people, but they tended to not save Muslims. That's the only group that they, on average, had singled out as not saving. Why? Well, there's a long tradition of the Crusades and anti-Muslim sentiment, especially from largely Christian denominations. And we wrote a paper about that. It shows up in a published book out of Temple Press about essentially how religion impacted who people would save. So I thought that was kind of an interesting finding here. Now, the whole point of this is not to say, these people are good or these people are bad. What we did was we exposed them to an experimental treatment. We forced them to choose between two people who had randomized characteristics. And over time and through statistical analysis, we have an experiment where they were forced to choose who they would save, and we get big, broad patterns from experimental data. It's called conjoint experiment. It doesn't really conform to what we would think of as an experiment per se. We didn't randomize who was being subjected to it. They all had to take it. Where we saw the randomization was in what kind of treatment they got. And then the statistical process allowed us to filter out and to control for these different types of variables so that we could see where there were patterns and not. And there are a lot of statistical procedures that you can do that can solve some of these setup concerns, or research design concerns, but there's very, very little you can do if you have a very bad setup and you have introduced bias. It's really, really hard to overcome that. So you have to be cautious, let the question drive the method. Now, there's so many different ways that you can do experiments, quasi-experiments, non-experiments. It's all about control and comparison and manipulation. So you can do a post-test only non-equivalent group. So you take an observation of somebody who's already gone through the treatment, and you compare it to somebody who has not gone through the treatment. That's a post-test only non-equivalent control group. If you don't have a control group, you're just measuring somebody who's gone through the treatment after they've gone through it. You've not measured them before. So then we could fix that with a pre-test, post-test, non-equivalent control group. How does that happen? Well, we have a pre-test and a post-test for somebody, but we have no control group. Or a control group that was not tested before. We only test them in the outcome. So we have like a pre-test, post-test for the people who've been subjected to the treatment, and then we measure a post-test for a control group, but we don't know what the pre was for the control group. That's also a different way to do an experiment. We could do a one-group pre-test, post-test, just pre-test, post-test. We could do time series. We could take numerous measurements over time to see how patterns trend on one individual or one set of individuals. We could take a cross-sectional developmental. In other words, we take a big group of treatment individuals and subject them to the treatment and see how it works over time. Time series is usually like one or two individuals, but a cross-sectional is lots of different cases. We could do longitudinal developmental. In other words, we can get lots of cases over time and see how they responded to treatments. There's just so many of them, but the whole point about experiments and quasi-experiments is control. Who do we compare it to, and how do we control external variables? Who do we compare it to, and how do we manipulate treatment? Okay, if you haven't learned this already, the question should drive your research strategy. Every one of these setups has a significant problem. There is no best method. So that's why you have to think about what you want to ask. The question then determines how you go about solving that. Now, just because they all have problems doesn't mean you can't overcome the problems. It just means you have to be aware of them and find ways to try and mitigate those problems. You have to choose what you think is best. You have to figure out ways to overcome the problems for every one of those things. And ultimately, this is kind of like the secret about a graduate level research methods class, is that you become the expert. Do the legwork, figure out why one thing will work and why one thing won't work, and think through all of it. Otherwise, you have to rely on somebody else saying, well, I'm the expert and I know best. Well, okay, that's fine. But by the time you finish your program, you're going to have a master's degree, and you will be able to stand up among all those other experts, given your experience, your training, all those other things. And you should be able to, go toe to toe with some of these people and say, well, why didn't you control for this? Or, what sort of things? And you should also be able to subject your own work to that same sort of criticism. When somebody asks you, well, why didn't you do it? You can't like go home and cry. You say, well, I thought about this and I thought about that, and this is how I mitigated this potential problem. And it's not the best way, but it's the only way I could figure out how to do it. And you start engaging in the process so that people can move science forward, and you can really try and figure out how to find these answers the right way. Experiments, quasi-experiments, non-experiments, that sort of setup is really, really crucial to a lot of the work that gets done. But you have to understand the underlying potential problems there so that you can engage in the work in a meaningful way.